glad to, well, I'd like to first ask you to join me in thanking the organizers and the presenters for a remarkable 2 days. My task is to summarize roughly 30 different presentations and put them in the context of a literature of over 9,000 publications. So, since I know you are all a kind and generous group of people, I hope you will be tolerant of a few stumbles. In the mid-1970s, interest in this illness resurfaced, spurred by research done here at NIH and in Denver and in Atlanta. And at that time, we had an illness that was characterized by only symptoms, pardon me. Can someone help me get back into show mode? Thank you. We had an illness characterized only by symptoms, no consistent objective abnormalities that were noted then. No physical exam abnormalities, no diagnostic tests, no proven treatments, no information on prognosis, no evidence of underlying biological abnormalities, nothing. It was a suspect illness. But that was 30 years ago. Where are we today? I'd say we have come a very long way. Can you help again? We've come a very long way, and we still have a long way to go. What I think has become clear over the last 30 years, and you've heard a lot of it in the last 2 days, is that cases do differ from healthy controls, and in some cases from disease comparison group controls, with regards to central and autonomic nervous system, metabolism, particularly energy metabolism, immune phenotype and function, the microbiome, gene structure, gene expression, and some epigenetic differences. The questions going forward are what caused the symptoms of the illness, what caused the abnormalities that I've just referred to in the various systems, what links together these abnormalities in these very different systems? What triggers them? And is there a hypothetical disease model that fits with the abnormalities that have been reported? So my agenda for this talk is to summarize the data presented at this conference, sometimes putting it in the context of the prior literature, regarding these various abnormalities that have been reported, and then to consider several theoretical alternative models that might explain much, if not all, of the data that we've seen. So let's start being as brain-centric as Dr. Koroshet's with the neurologic changes, which involve structural and functional brain imaging changes and autonomic abnormalities. The past literature has found hundreds of studies that demonstrated different kinds of neuroendocrine dysfunction. Many studies have demonstrated cognitive problems. Many autonomic dysfunction, you've heard a lot of that over the last 2 days. MR studies, both anatomic and functional MR, SPECT, PET, EEG, all demonstrating something is going on in the central nervous system. You've heard several people refer to this paper by Nakatomi from several years ago, the first serious evidence, in my view, of brain inflammation in patients with this disorder. Small study, hasn't yet been replicated, but the distinction between cases and controls was nearly perfect. As you can see, there were higher levels of a signal that measures an translocator protein that is a signal for activation of glial cells in the central nervous system. MR spectroscopy of the brain, very recently reported by Jared Younger, was a study of 15 women and with ME/CFS and 15 matched healthy controls, and found, as Jared reported, abnormalities in multiple brain regions, particularly the left anterior cingulate, metabolite ratios in 7 regions that correlated with fatigue, and an increased ratio of choline to creatinine and increased lactate being particularly prominent findings. As Jared pointed out, those findings of increased lactate were just the latest. This is a highly replicated result. This is one paper from 5 years ago comparing patients with ME/CFS to patients who were healthy and to patients with generalized anxiety disorder. Very clear and dramatic differences in lactate levels. Peter Rowe, in talking about autonomic things going on in the brain, talked about CBF, cerebral blood flow reductions, being very substantial, and showed these data. He just showed them again a few moments ago. Even in patients undergoing upright challenge, orthostatic challenge, who had a normal heart rate and blood pressure, there was a clear decrease in cerebral blood flow, both initially and then an increasing decrease towards the end of the tilt challenge. Metabolic changes. We've heard a group of different metabolic changes summarized at the conference. Impaired ATP production, general hypometabolism, and I would argue, even though we didn't present it at the conference, oxidative and nitrosamine stress. At least my summary of the early papers on metabolomics in ME/CFS is as follows. Most of the studies have found that levels of most metabolites are lower. As Bob Navio was first to point out, is similar to what happens in the state called dower in the worm, C. elegans, or in hibernation in mammals and some birds. We'll come back to that in a little while. Abnormalities incriminate impairment of cellular energy and oxidative phosphorylation, indicate a possible defect in one particular enzyme, pyruvate dehydrogenase, that has not yet been replicated, to my knowledge, and defects in pathways converting sugars and lipids and amino acids into energy, into ATP. There is some evidence linking these metabolic changes to a state of chronic inflammation or activation of the innate immune system, triggered perhaps by NF-kappa B, which is measurably higher in the patients in studies that have looked at it, which upregulates COX-2 and INOS to generate both oxidative and nitrosamine stress, the consequences of which is to damage nucleic acids and lipid membranes, which should be damaging to mitochondria, which should lead to an increase in anaerobic metabolism and increase in lactate. And I would say that the literature is generally consistent with this model. Maureen Hansen at the conference pointed out that we should be looking at the metabolomics not of populations of cells, but of individual cell types, and found quite different results when she looked at CD8 T cells as opposed to CD4 T cells with different effects on mitochondrial respiration and glycolysis. Nancy Klimas argued that metabolomics data can be merged with drug-gene interaction data to identify potential therapeutic targets in this disease. So beyond simply looking for a metabolomic fingerprint that might be diagnostic of the illness, metabolomic data may also point the way to some successful therapies. I believe this is the data from Uppsala, spinal fluid protein metabolomics, finding clear differences between ME/CFS and matched controls with generally increased levels of markers for neuroinflammation, cell damage, and repair in the people with ME/CFS. Both significant similarities and differences were found when the ME/CFS people were compared to those with multiple sclerosis. There were some apparent similarities and some clear differences. Bhupesh Prusti talked about whether the energy generation mitochondrial abnormalities that data indirectly suggest are present might be caused by chronic viral infection, and mentioned experimental evidence that one particular virus, human herpes virus 6, in one cell line, active infection, not latent, fragments mitochondria and inhibits the ability of mitochondria to fuse and repair, and reduces ATP production, not surprisingly. And also reported that serum from ME/CFS patients fragments mitochondria, and removing that serum resolves the fragmentation. Mechanisms not yet pursued or uncovered, but a striking finding, particularly given Ron Davis and other people's findings, that there are some molecules in serum that cause various aberrant physiology, and we need to figure out what they are. Ron Davis presented this slide. I'm waiting for the paper, which will be published, as I understand it, in PNAS in the coming months, that found one abnormality that clearly distinguishes patients, in this case 20 patients and controls, perfectly distinguishes them from healthy controls. That also obviously is a clue as to some underlying biology that could be causative of the symptoms of the illness, and I'm sure that's going to be pursued. Immunologic changes. I've always been confused about immunology, but everything fell into place when Jose Montoya demonstrated this highly simplified overview of human immunology. Enough said, I'm still confused. But in the immunology of the 20th century, that long time ago, it was already clearly established. I would argue by the turn of the 20th, 21st century, it was clear that in patients with ME/CFS, there were increased levels of circulating immune complexes, increased levels of immunoglobulin G, decreased levels of certain IgG subsets, increased numbers of CD8 cytotoxic T cells that bear various activation antigen markers. That NK cell function was impaired, and that there were increased blood levels and increased lymphocyte production of pro-inflammatory cytokines. Since that 20th century immunology, 21st century immunology has filled out picture. Here was a study, the first study from Ian Lipkin's group, finding that blood levels of many cytokines are significantly higher in people with ME/CFS than in healthy controls, but only in the first years of illness, not thereafter. It is as if an initial immune system response to some perceived threat tries hard, several years, and ultimately becomes exhausted. And if anything, the Lipkin group finding was that after 3 years, the levels of many cytokines started to dip a little bit below those in healthy controls, as if the cells making those cytokines had been exhausted by the effort. Levels of many cytokines in spinal fluid also were found to distinguish patients from healthy controls in another study from the Lipkin group. And then finally, in this study that Jose Montoya presented at the conference, levels of many circulating cytokines were found to correlate positively with the severity of symptoms. Important because if you find a biomarker that appears to be abnormal in people, you would like to know whether that's an epiphenomenon or whether that biomarker actually contributes to the suffering, to the symptoms of the illness. And several of these cytokines seem to correlate with symptom severity. Oh, I see. Jose Montoya also pointed out that many of the cytokines that he found to be differentially upregulated in people with ME/CFS are made by mast cells. That's important because, particularly in the last 2 years, there's been a growing body of evidence from studies as well as clinical experience suggesting that some patients with this illness may have a mast cell activation syndrome. That we need to understand that better, but there are both diagnostic tests and treatment implications in establishing that connection. Jose also reported at this meeting some HLA associations. These are HLA genotyping, not the old-fashioned and less reliable and precise way of HLA typing, finding several markers that were associated with the illness itself, at both increased risk and decreased risk of the illness, but also markers that were associated with increased and decreased risk of severe illness. So separate markers correlated with the illness as well as with illness severity. That's new information that may be a clue that needs to be followed up. Durya Unemans pointed out that the CD4 and CD8 T cell subsets are different between patients with this illness and healthy controls, particularly in the older but not younger groups. That's curious, and the reason for it needs to be pursued, but certainly when this phenomenon is looked for, age needs to be controlled for, as data shows. This is Mark Davis's data showing that he finds an increased number of clonal CD8 cells responding in ME/CFS patients compared with controls. What is less clear at this point, and what he's obviously pursuing, is trying to identify the epitopes against which those clonal CD8 cells are targeting, because that could give a clue as to what the trigger of the illness is, at least in those patients. Epigenetic studies, the past literature has found differences in people with ME/CFS compared with healthy controls with regards to the DNA metallome with regards to the expression of microRNAs, and we'll come back to the data at this meeting on that subject. Differences in transcription factor levels, particularly NF-kappa B, and increased HDAC expression, all different kinds of epigenetic controls of gene expression. This is from Moreau's report at this meeting. He has been looking at various microRNAs in people with ME/CFS versus healthy controls, and has found a whole group of microRNAs that are differentially regulated in the illness compared with health. Some of them also correlate closely with the symptoms of the illness. This example, microRNA 155P correlates with mental fatigue. Moreau also found that when you just let the microRNA data define subgroups, independent of any clinical information about those patients, what you see is that the subgroups defined by the microRNA profiles also have characteristic and very different clinical profiles. The symptoms, for instance, in subgroup 1 are very different than the symptoms in subgroup 5. Provocation studies of several types. Why do provocation studies? The rationale is pretty clear. Biomarkers seen in resting people with ME/CFS may or may not have anything to do with causing their symptoms. So if there are things, challenges that make people's symptoms worse, it would be nice to know if they also make the biomarkers worse, because if they do, it would suggest a connection between the biomarker and the symptoms. And that's the rationale for doing it, as well as that the key symptom of post-exertional malaise has an underlying physiology that needs to be understood, and obviously cognitive and physical challenges are the way to elicit post-exertional malaise. Past studies of exercise testing in particular are numerous, and I would argue quite consistent and well replicated. There are abnormal responses in people with ME/CFS to anaerobic threshold, to heart rate, to VO2 peak, and to peak work. There is also an impact on pain. The pain threshold in people with ME/CFS is different at baseline before exercise, as you can see, blue versus blue being ME/CFS and the orange being healthy. But notice what happens after exercise. The threshold goes up, that is the pain goes up in the ME/CFS patients, but goes down in healthy. So that's the phenomenon of central sensitization, and it has been reported by many investigators in this illness. Upright challenge also affects cerebral blood flow, as I showed a minute ago, and as Peter Rowe demonstrated at this conference. This response to orthostatic challenge, as Peter pointed out, has certain pretty consistent findings and some quite inconsistent findings. There's venous pooling, particularly in the blood, up in the legs. There is diminished vasoconstriction, which is normally the protection against venous pooling, diminished intravascular volume. But at the end of this process, this pathologic process, process, people respond quite differently. Some with orthostatic hypotension, others not. Some with neurally mediated hypotension, some with POTS, and some without any significant heart rate or blood pressure drops. That variability needs to be pursued with physiologic studies. Betsy Keller described many studies demonstrating chronotropic intolerance, defined as on exertion, an unusually low heart rate, low ventilatory rate, so there's less carbon dioxide being thrown off by the body, and a lower blood pressure. And she said that her 2-day studies, exercise challenge studies, found that this was a much more dramatic abnormality on the second day than on the first. Another reason why the 2-day study, in my view, has real value in both understanding the illness and unearthing the underlying abnormalities. This phenomenon is probably a manifestation of autonomic dysfunction. Hard to think what else it might be, but more formal study of that should be done. So, putting it all together, the central and autonomic nervous system abnormalities, the metabolic abnormalities, immune system, epigenetic differences, microbiome differences, which we'll come to in a minute, and the provocable abnormalities, the various provocation studies. Are there any models that would explain it all or unite it all? And there may be, and I'm going to talk about several which other speakers at this conference have alluded to. I would say there's 5 models that seem to me most intriguing. One that we didn't talk about at this conference at all is really been raised by Ned Sharpless, who's the head of the NCI here, who in cancer patients has a lot of data suggesting that excessive cellular senescence leads to the generation of fatigue-inducing molecules. I had a conversation with Brian Wallet and Ali Nath that they will probably be looking for these molecules in the studies that are being done in the intramural program here. Sickness, behavior, inflammation is another model. Come back to that. The Dower hibernation torpor model is another model. The cell danger response incomplete healing model is another model. And then the microbiome as a driver of abnormalities that lead to the symptoms of this illness is yet another model. I would argue that at least the last 4 will be linked with each other and maybe different expressions of the same ancient physiological response in animals. I'll come to why I say that. So the first model is the sickness behavior inflammation model. Jared Younger referred to that this morning. What's that mean? What is sickness behavior? What do you feel like when you get sick, when you get the flu? Everyone has had the flu and everyone has felt sick. One phrase that's often used is, I feel like a sick puppy. We use that phrase because when our pets get sick, we see their behavior, we see the parallels between their behavior and ours, and we feel like a sick puppy. And that's what a sick puppy feels like. And that sick puppy is experiencing the same set of physiologic changes that we experience when we get sick. Sick acutely, the sickness behavior response is a evolutionarily preserved response in virtually all animals in which it's been studied. That when there is illness, it's been studied most in infectious illness, but not exclusively, there is a temporary response to that injury or infection in the animal. purpose to that response. The purpose is to focus the body's energy stores on fighting the infection or the injury, healing the injury, generating acute inflammation, fever, and the sickness response also has the brain discourage all sorts of behaviors that consume energy so that the energy can be saved. So people are lethargic. They don't move around a lot and spend energy that way. They're socially withdrawn. They're achy and they don't want to move too much. They're sleepier and they sleep more. Loss of libido, difficulty thinking, depression, anorexia. These are all manifestations of the acute sickness response. The question raised by many people is whether this acute sickness response, something we are all biologically wired for, is in some people somehow made chronic, unable to turn itself off, and whether that is what's happening in ME/CFS. Here is a model. It starts with the brain, and it postulates that in the brain there is a fatigue nucleus. There is some group of cells that perceive and monitor fatigue. There is evidence that those cells may be in the basal ganglia, illustrated here, or in the prefrontal cortex or in the anterior cingulate. It says that activation of the brain's innate immune system, the microglia, prominently, not exclusively, yields cytokines that trigger the fatigue nucleus to feel the experience of fatigue. And the question is, what activates the brain's innate immune system? Jared Younger listed a group of things earlier today: infection of the brain, directly of the brain, autoantibodies in the brain, toxins that reach the brain. He didn't mention it, but there's also data that the state of obesity, chronic stress, and elevated levels of leptin, whatever causes leptin to rise, all activate the innate immune system of the brain. Also, infection or inflammation elsewhere in the body outside of the brain can signal the innate immune system of the brain to turn on. It's essentially the periphery of the body fighting a battle, the immune system, and saying to the brain, hey, we've got a war on down here, get ready up there to do something. How are those signals from the periphery delivered to the brain? One way, known about for several centuries, is the humeral route of blood that flows through a porous blood-brain barrier and with molecules and cells that reach the brain that normally shouldn't be able to get there. This is not a new concept that the blood-brain barrier can be breached. Cajal first observed and drew it more than a century ago. This is his drawing of cells penetrating the blood-brain barrier. What's more interesting, and only in the last 20 years has become solid, and Jared Younger and Mike Van Elseker referred to it, is that peripheral inflammation also signals the brain, not by blood-borne signals, but by retrograde transmission of signals up the vagus nerve to the brain. So look at the bottom figure. My pointer isn't working, but look at the bottom. Infection outside the brain triggers the innate immune response. That generates neuroexcitatory molecules that are picked up by chemoreceptors on vagus nerve ends throughout the body and send signals up the vagus nerve. Glial cells that are enveloping the vagus nerve, as Michael Van Elseker postulated several years ago, themselves can become infected and send those same signals up the vagus nerve, where they arrive in the nucleus tractus solitarius and send signals to the rest of the brain that consummate in what we call sickness behavior, the changes in behavior that manifest as sickness. The potential sites of peripheral inflammation I basically just summarized. One possibility suggested by Peter Rostock today is that the phenomenon he described of neurodynamic strain might also be sending retrograde signals up the vagus and activating innate immunity in the central nervous system. The Dower hibernation torpor model. Dower in the worm C. elegans is a protective effect state that's temporary, and when the organism is challenged by harsh environmental conditions, that can be temperature, absence of food, whatever, crowding, but when the signal, the distress signal, is received by the worm, it goes into a state where it shuts down most of its energy requiring reactions to preserve the energy it has to contend with the vital threat that it perceives. In other words, it makes physiologic changes that improve its odds of survival, but badly compromise its functionality. The same thing exists in hibernation in mammals. Food shortage, temperature change, trigger protective responses that help the animals preserve energy through the energy needed to survive through rough times. The thing that I hadn't realized until reading this literature is that the state of hibernation in mammals is not a semi-permanent one. The bears don't fall asleep in December, wake up in late March, and never awaken in between. In fact, those states are quite transient. They wax and wane, and there are signals that cause animals to enter into the states and exit from the states of hibernation. Torpor can even occur in warm temperatures, estimation in some animals. I make that point because I'm arguing, obviously, that the cyclic depression of function in ME/CFS has some parallels with the cyclic process of Dower and hibernation and torpor. Both Dower and hibernation are regulated by the same transcription factors, and those same transcription factors also are heavily involved in the stress response and detoxification of reactive oxygen species and innate immunity. They both involve increased glycolysis and decreased aerobic respiration. Here are the parallels with ME/CFS. They may involve